Okay, so the addition, deletion and substitution have the same effect of point mutations that result in the synthesis of different proteins, which can have different functions. So the changing of the RNA, changing the genes. If you remember the circle that we had before to code for an enzyme or a protein, multiple 3 bases can code for the same protein, but one base does completely change can completely change the protein that is produced from the mRNA strand. So it can have different functions, or it can have the same function, but this addition, editing, deletion and substitution does increase the range of proteins that can be produced from a single mRNA strand. So there's following are the mechanisms that regulate the process of protein synthesis. So protein synthesis is quite a long mechanism, which I will go through in another video, but there's a few mechanisms that regulate this process of protein synthesis. Firstly, we talked about degradation of the mRNA in the cytoplasm before, that the cap and the tail does delay the degradation of the mRNA in the cytoplasm. The more resistant the molecule, the longer it will last in cytoplasm with the cap and the tail, for example, and therefore the more proteins can be synthesized, so it's just more effective and more of a faster rate. Secondly, the binding of inhibitory proteins to the mRNA. So when you bind inhibitory, I'm spelling because I don't know, inhibitory proteins to the mRNA strand, this prevents it from binding to the ribosomes. So it prevents the mRNA strand from binding to the ribosomes, preventing any synthesis of proteins. So you can just have inhibitory proteins that don't do anything, preventing the mRNA strand binding to the ribosomes, preventing any actual synthesis of proteins that do anything. And thirdly, the activation of initiation factors. So activation of initiating factors. So activation of initiating factors. These aid the binding of the mRNA to ribosomes. It does the opposite of binding to inhibitory proteins. So activation of initiation factors actually aid and forward the binding of the mRNA to the ribosomes. The eggs of many organisms, for example, produce large quantities of mRNA, but it isn't required until fertilization, at which point the initiation factors are activated. So, protein kinesis are enzymes. These protein kinesis enzymes catalyze the addition of phosphate groups to proteins. So this is just a bit of an example. So protein kinesis, K-I-N-A-S-E-S, protein kineses are enzymes. These are enzymes, and catalyze the addition of phosphate groups to the proteins. This therefore changes their tertiary structure, and therefore it changes the function of the protein. Just by an enzyme such as protein kinesis adding a phosphate group changes the tertiary structure, which in turn changes the function of the protein. Protein kineses are important regulatories of cell activity. They are often activated by CAMP. Post-translational control, as we see here, involves modifications to the proteins that have been synthesized. Post-translation control examples include So after translation, you can modify the proteins. So after translation, proteins are modified by 3 ways. 1, the addition of non-protein groups. Addition of non-protein groups. So examples of these are carbohydrates, lipids, phosphates, anything that's not a protein, and adding that to the protein groups. Secondly, post-transcriptional control includes modifying the amino acids and formation, actually modifying the amino acid and forming bonds such as disulfide bridges. So disulfide bridges. So these are more bonds. So we've got here addition of non-protein groups, carbohydrates, phospholipids, lipids. And then secondly, we've got modification of the actual amino acids. So disulfide bridges. And thirdly, the modification of the CAMP. So the CAMP is important because it actually is the activation of the enzyme, such as the enzyme of protein kinases. So the modification of the CAMP, for example, in the lacoperon, which we've talked about before, the CAMP binds to the receptor protein, increasing the transcription, or actually allowing the transcription of the 3 structural genes LAC-Z, LAC-Y and LAC-A. So the CAMP is originally bound, and the removal of it allows the translation to occur. So this is the Homobox genes. So a Homobox genes, as you can see in a box, a clever idea, are a large family of similar genes that direct the formation of many body structures during the early embryonic development. So these are similar genes that direct for the location of the structure. So direct for location of the organism's structure. And this happens during embryonic development. So this is very important because embryonic development is when you're actually forming the shape of the human, the animal, whatever it is. So these Homobox genes have polarity, etc., etc., and are the same genes, or similar genes, just direct for the location of which body parts. Homobox genes, or cell adhesion molecules, which you remember from Cameron, CAMs. So CAMS are cell adhesion molecules. Cell adhesion molecules position the cell in a multicellular organism is influenced by the contact of these CAMs with other cells and the extracellular matrix. So it sounds confusing, but basically CAMs position of the cell in the organism is basically influenced by the other cells and the matrix, extracellular matrix. So the Homobox is slightly different to something we're going to later talk about, the Hox genes. The Homobox genes is the group of genes which all contain a homobox, which is a selection of DNA, as you can see in the colours here, the selection of segments of DNA, 180 base pairs along the coding of 60 amino acids. The domain binds to the DNA and switches the genes on and off. The Homobox, therefore, are regulatory genes. So these Homobox genes are regulatory genes. Homobox genes are regulatory genes as they are able to switch the genes on and off. The genes help with evolution. So these idea of the Homobox genes help with evidence for evolution, as the same Homobox genes are in every organism. They have identical nucleotide sequences. So you can also see that the animal is segmented, which is basically every animal ever. You've got an insect here, but it's not just always an insect, it's a human. We have segments, head and tail. So it helps with evolution evidence as it's segmented and it has the same identical nucleotide sequences. So we get to something called a Hox gene now. A Hox gene, slightly different to a Homobox gene. Hox genes. They're one group of Homobox genes that are only present in animals. So while Homobox genes are present in everything, Hox genes are only present in animals. Only present in animals. And it is the positioning of the body parts, and these are found in clusters. So they are positioning